So good morning, everyone. My name's Ed Hookway. I come from the very long-winded Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences at the University of Oxford. And I've come to talk to you today about not so much the detail of the research that I do. I don't know if any of you were at the previous conference in Bristol, it was, and heard what I said, but more about sort of the day-to-day life of what we get up to as a scientist, because I can imagine for many people it's a black box where you really have no idea about what it is we actually do. So I thought that is sort of more what I'll focus on today, and then touching on the areas of chromosomes, cells and computers, which pretty much sums up what I do for my research. I think probably adding a fourth C to that as well of conferences would be appropriate, because over the last couple of months actually I've been going and talking about the bone cancer research we do in Warsaw in Poland, in Vienna in Austria, and I'm also skiving off actually from a different conference today in Oxford, for which I'm talking tomorrow, to come and speak to you here. So just a little bit about myself, as I'm to know where your money's going. So I'm a physician, a doctor by training. I trained in Cambridge and then worked in the NHS for a number of years. My clinical interest was always in oncology and cancer, and I spent a lot of time working in departments for oncology, so in Cambridge and down at the Marsden in London. But then I sort of made the decision to have a slight change of track and go in and focus on sort of doing basic science research. The term basic research is maybe misleading because there's nothing basic about it. It's the sort of the fundamental research that underpins why cancer happens and what's going on. And this is what we need to understand, to learn. Why is it that some people have normal bones, and why is it that some people have bones which develop cancer? So working where I do in endorms, as we call it, it's a great sort of multidisciplinary environment, because we're a whole department focused on bones and muscles and musculoskeletal things. So we have a lot of expertise there, not only in cancer itself, but in everything to do with sort of bone biology and bone oncology. My own research sort of falls into two very different strands. The first thing I do and look at is to do with how bone actually gets eaten up, so what we call osteolysis. One of the things that we know in lots of primary bone cancer is that we get a sort of a destruction of the bone and a weakening, and trying to understand how that happens is a tricky job. If you actually take cancer cells themselves and put them onto bone, they won't eat it away. It requires recruitment of other cells in, which actually break down the bone. And we're trying to understand the interaction between the cancer cells and the cells which do the breaking down. Now, to do this, we've got some sort of very complicated tools, I say jokingly. We have a big slice of an elephant's tusk, which was confiscated from someone at Heathrow by customs, which we use as our model of bone. And so I spend a lot of time sitting with a circular saw, a hammer and a punch, chopping up bits of bone to get very small, very flat surfaces, so that we can actually look and see what is eating the bone away. And we've just had to spend about £400 to buy a new special blade for our saw, just to make sure it's sort of really flat. And I sort of see myself working mainly as a molecular biologist, somebody who works sort of down in deep, sort of the complex things. And so as I'm sat there with my saw and my hammer and hitting things, causing a general nuisance in the lab to everyone else, it just goes to show the sort of the very different aspects that we're taking. Yes, sir?
Can I ask a question?
Yes, certainly.
The elephant tusk and
Yep.
our bones, are they obviously very similar then?
Yeah, so bone is pretty much bone. It's the reason we use it is because once you've managed to get hold of a tusk, which involves lots of paperwork, lots of inspections and licenses, because of course all sort of the ivory trade is illegal, it is enough to last for years and years and years. So we just take very, very small thin bits off it, and we sort of get a couple of millimeters across, and we can put things on it and then have a look to see, has the bone actually been eaten away? So the difficulty is we can't really do that with human bone because we don't have the access to it. So it's a well-established model for using. Okay? I think if anyone has any questions, do feel free to chirp in. This is more a sort of a discussion interactive session, I hope. So it's, you know...
If you get offered my son's an amputee, wouldn't the leg he lost, wouldn't that be beneficial to you?
So from... I think this is going to come on to what we talk about a little bit later when I sort of talk more about the cells we deal with. So for us to understand what's going on, we try and sort of grow cancer in a dish. And that's where the cancers which have come out of people become very useful. The difficulty is with it is most people before their operations have chemotherapy, which kills off the cells. And you know, if it's an osteosarcoma, then you want 90 to 100% of your cells dead, which is why we get the very sort of intensive chemotherapy, which is great for the individual patient. Unfortunately for us, it's not so good. So I spend a lot of my time...
Some people leave the bodies to research or whatever, yeah. People,
I mean, I think it's a difficult area. I think, unfortunately, you know, we all know how horrible bone cancer is, and if somebody's been unlucky enough to lose their fight with it, they've probably had a lot of treatment along the way. And what we want to try and do is try and get a representative sample of it to begin with, because that's the sort of the real entity that we're fighting. And I think it's just very difficult when people have gone down the line, and especially it's the sort of thing that you don't want to be worried about scientists coming and poking your bones when you've got more important things to be worrying about. Yes, sir?
So when our little girl went to Birmingham and had a biopsy, once they diagnosed the osteosarcoma, do those biopsies ever get passed on to you, or do they use them for their research?
So the biopsy is a very interesting question. So I spend a lot of time sort of running around Oxford with dishes of things which have been biopsied. So the difficulty is that the most important thing for a biopsy is to get all of the clinical information that's needed. And the biopsies, I don't know if you've ever seen them, are tiny, tiny, tiny. And because you don't want to go and take a big lot out, so it's very small. And what we tend to find is that the time it takes for the processing of the biopsy specimen, and I work a lot in Ewing sarcoma, where there's a sort of a characteristic chromosomal translocation, and that takes a couple of days for the NHS tests to work through. And I can only get samples at the end of that, because what we don't want to do is to have a big chunk come to me to do research on, and then have to go back to the patient and say, I'm really sorry, we need to do another biopsy because we haven't got all the information. So the answer is I try to do it. And
one for you, one for the... they're really small.
So we try to do it, and as an example, I've had sort of 4 Ewing sarcomas from biopsies that I have tried to get growing so I can investigate further. And of those, 1 has started to grow, which is very good for me, because we need these tools to investigate. And when you have a cancer which is effectively growing in a dish and just will keep growing, I can do as many different tests on it as possible. And so that's the ultimate aim. The other 3, we were able to do a one-off experiment and say, these are fresh cells out of someone. we had a particular theory we wanted to test, we were able to give it to them and get the right answer, which is very good for us, but as Ewing's and all bone cancer is so rare, it's very difficult to do your research based on waiting for something to come along and do one test. So it's tricky, and I think what we've got to remember is that it's the best interest of the patient is paramount for us.
So why do people get bone cancer in your research? What's the...
So it's a really tricky question. I focus on an area of research which is called epigenetics, and this is where the chromosomes come in. So epigenetics is a sort of a buzzword that's starting to go round in science circles, and the easiest description of it is if you think of two identical twins, and if they're identical twins, they have exactly the same DNA by the very nature of them being identical. And the DNA is the information inside you which codes for all of your genes and your proteins. And traditionally in cancer, we think of a mutation in the DNA as being the cause of the cancer. So there are some sort of very famous examples of proteins which in cancer we know get mutated. But when we think about identical twins, why would one of them get cancer and one of them not? And one of the possible explanations for this, and by no means I'm saying it's the only explanation, is to do with the difference in the way that the DNA is sort of controlled and expressed within the cell. And we call this epigenetics. So it's changes of gene expression outside of changes of the DNA. And the way of thinking about it is that there are some, when you have your gene, you can either express it to a normal amount, which is what you want, or it could get turned off and silenced. So if you have something which is vitally important within your body, which gets turned off, very important for protecting against cancer, and it gets turned off, you're opening up yourself to the risk of cancer. And this is something that we found in Ewing sarcoma, one of the sort of very important genes for detecting when there's a problem within the cell and sort of saying, hang on, something's wrong here. And normally if a cell gets that message, it decides to kill itself, because it's better for the cell itself with a potential problem to die than for the mutations to hold and to expand. And one of the proteins involved in this process effectively gets shut off in Ewing sarcoma. And we're looking at ways of actually telling the cell, you need to be expressing this protein, and to tricking it into re-expressing everything so it can fight the cancer itself. So it's a very different way of thinking about sort of traditional chemotherapy. In traditional chemotherapy, we are effectively poisoning the body, and we're hoping that the chemotherapy targets the cancer cells more than normal because they're dividing rapidly. And
is that epigenetic factor that you're talking about, is the one that is related to the effect that, for example, mifamortide have had on osteosarcoma, as in helping the immune system to detect and delay it?
So it's a sort of a different kettle of fish to immunotherapies. So in immunotherapies, you're trying to get the body's own immune system to kick into effect and to recognise the cancer. It's a similar idea though, in that what we're trying to do is to do something which specifically targets the cancer. So, I'm sure a lot of people here have lived through, if not themselves, and seeing their loved ones undergo chemotherapy. And it's horrible. Yeah. And trust me, as the doctors who give it, we don't really like it.
Barbaric.
It's horrible. And, when you, especially with young kids, when they come in and you have to say, how awful you felt last time, well, here we go again, and it's going to get worse. What we want is something that's specific. And by finding these changes within the cells, which are specific to the cancer cell and not specific, and your normal cells don't have the problem, the cancer cell does, and finding a very specific way to sort of flip it back into controlling itself, then we hope to be able to give really specific targeted therapies.
Because chemo has been around for a long number of years, my oncologist told me that the development of chemo is about the end. Chemo is not really going to develop that much more. They're going to do it what you're doing. Is the idea is to go back to the beginning.
So I think the what we call the cytotoxic chemotherapies,
Yes.
It's going to kill everything.
Yeah.
I mean, a lot of them, or some of them at least, were developed out of the mustard gases of the First World War.
Yeah.
And, we see unfortunately how good they are at killing people, and that's what you want to do. Or that's what they had to do. You wanted to give enough to kill everything off that was bad, and hopefully leave the person intact. So, I think it's difficult to see how we can go further along that line without sort of having the toxicity associated with it as well. So it's trying to find these specific molecular targets that we're after.
And do you ever think it will go to the point where, in the future, where injection, like you get an injection for diphtheria as a child?
So cancer vaccines, yeah. So
it's a long way off, but you think that might happen in years to come?
So we have wandered into one of my very many areas of ignorance here. There are people far cleverer than I am working on this, and so they clearly think there's something in it.
Yeah.
But I'm afraid as far as the details go, I'm not your chap for that.
But it is something they are looking at.
Absolutely. And it's like the immunotherapies that we mentioned. It's getting your own immune system to fight.
Can I ask you about the targeting, because it obviously sounds very exciting and very sensible. And it's a difficult question, I know, but how close do you feel that you are in making some sort of breakthrough? Because the idea is great, and it's that translation between the idea and what do you need to do, and it's probably the rest of your talk here, in translating it into some sort of breakthrough. How does that happen?
So we think, as far as in a dish goes, we are hopefully about to publish something which is a breakthrough. No, it's not entirely a secret.
So we just need to take all the cancers and put them in a dish, and we're okay.
So this is sort of looking at a variety of cancers affecting bone. So the Ewing sarcoma specifically, also multiple myeloma, which is a disease which affects bone. We have discovered a mechanism, we hope, which seems to specifically kill these cells, whilst if we give the same drug to normal tissue, as far as we can see, it does nothing. Now, what I have to say there is that this is in a dish, in a lab.
So how long is that from there to reality?
Years,
Oh, yeah.
unfortunately. And the difficulty is that the compounds we're using are very good in our dish, but they would be no good. I couldn't give it to you as a tablet or an injection. So what we are doing is we're trying to understand the fundamental reason why these things are happening. And then what we need is for people in industry, in the pharmaceutical sector, once we have shown to them, hey, this is a good thing, then they can start to target and develop the drugs. And, we need to be really cautious, because have been researchers for decades standing up and saying, we've got this great thing, which then doesn't translate. But it's the first time with this sort of specific kind of modulation within a sort of the Ewing sarcoma in the way we're modulating it, that is causing this sort of effect.
Anywhere else in the world on
Oh, our competitors are working on it and trying to publish it before we are. So that's certainly, sir.
Sorry for my son.
No, don't worry.
The lung sarcoma.
Mhm.
But let's say that the cancer cells which give the metastasis, especially for example the lungs, are they the same cells? Or is it mutated?
So it's a very interesting question, and there.
You can actually kill anything which is within the lung or in the area, but the main problem,
Mhm.
as far as I'm concerned, no, it's actually metastasis
Mhm.
and that's really.
So if we think about metastasis in general, there is evidence that actually they do mutate separately over time, and so they are ever so slightly different from the original tumour. We would hope, or I see no theoretical reason why it wouldn't work, but as I say, we are in a dish, and it's something that it's in theory it should still work.
You need another dish with the metastasis.
Well, unfortunately, it doesn't quite work like that. We need to. I mean, the next step we need to be thinking about is looking at this in animals, in animal models, and then those sort of questions can try to be answered. But as I say, this is all very early. Sort of, for me as a scientist sitting in my lab, this is really exciting. It's the difficulty in the translation and getting it through into the clinic, into people, which is just incredibly difficult overall.
When our daughter was first diagnosed, I remember on the news, is there an African rodent that never gets cancer, that's never developed a tumour, and they were trying to do the DNA code of that creature? Is that any further along the line? Or?
So I'm not familiar with that specific example. There's a myth that goes about that sharks never get cancer as well, which is not true. So I'm not familiar with that specific example. I would imagine that that is possibly not strictly the case.
Can I just go back to where you started and the bone samples? I think it was one of our conferences a couple of years ago, and we had a vet that told us that osteosarcoma
in dogs
was their biggest disease, and
Yep.
the second biggest in cats. Now, the great thing about vets, they don't actually need the same permissions that we do for humans. I mean, is that an area then that you've asked the veterinary sector to come forward with?
So it's not something I'm specifically involved with. It's very interesting because the osteosarcomas in dogs relates to the size of the breed of the dog. So it's the Great Danes, which are the sort of the very massive dogs, which tend to get it, and that's interesting as well. And it's why there's slightly more men than women get it as well. And is it to do with growth and height and actual turnover of the bone? Which would make sense, because if the bones are having to turn over and copy themselves, there's more chance of making a mistake, which is possible. So I think this is the sort of thing that at the fundamental level we're working at, the difference between human, dog, whatever, tends to be quite small.
Yes.
Because
that's an interesting point, because I think over the years, certainly noticed that many of the teenagers with osteosarcoma are very tall. All of a sudden. And certainly my son was like that. He was a titch beforehand, suddenly had a growth.
Well, and it's related to the growth spurt and the growing. It's something that would be great to understand.
Yeah.
I mean, I think this is just a perfect example of, we have this observation that several people here in the room have made independently, and we don't understand it. And the problem with rare cancers are trying to understand what's going on is all the harder because we don't get the samples and the specimens.
And just finally, I'd just ask you, do you regard what you're doing as a pathway to a cure or to a treatment? And thinking in terms of improving the taking of chemotherapy in whatever cocktail that might be.
So when I go to work, I don't think...
Hi, Edward, can you be wrapping up?
Yeah, let's wrap up. So I don't think, today I'm going to go and find a new treatment. I think I want to understand what is going on, which could lead others possibly to identify a novel area that can be attacked. So ultimately, yes, my goal is a new treatment, but I'm sort of working much earlier on in the process of trying to discover the things that we don't know about the cancers, which can give us more opportunity to target them.
Can I ask about, before we go, there's a lot of talk about tumour banking, and Andy Hall is keen on that.
Mm-hmm.
But clearly you'll be banking tumours that have had chemotherapy.
Mm-hmm.
And if you've got 98% of those tumours killed, is that worthwhile?
So tumour banking is definitely worthwhile. It's something we do a lot of in Oxford, and I know Professor Hassan spoke to you yesterday, and he's very heavily involved in the biobanking. So it's definitely worthwhile, because as our techniques improve, the 2% of viable cells left might become really important, and there are things, even though the cells are dead, there are important things that we can take from that.
The cells, the 2%, are ones that have survived and therefore quite valuable.
And why did they survive and the others didn't?
On that line of thought, this might sound a bit macabre,
Mm-hmm.
but would you benefit as a scientist to have healthy human bone banking? Because I'm a donor,
Mm-hmm.
and if I could say, okay, Edward is having all my bones when I die, as long as I'm healthy, do you think that will help you, or you're happy with your tusk?
It's difficult. I think we have access to normal bone in lots of other different contexts for research. It's too precious for us to use as our let's try and eat it away. But for other things, we can get hold of it to understand what's going on. So
we don't need a bone bank, basically.
No, it's just
Tumor
your-
bank,
it's just your
but not
bones.
the bone
It's just-
bank.
yeah.
Just my bones.
Even with his tusk.
Linking on with that,
Yeah.
now we have DNA mapping
Mhm.
and know where the mutations are
Mhm.
occurring. In theory, would we be able to then start tracking back in time, start looking at samples in the past
Mm-hmm.
to find a specific point where, hang on a minute, these mutations at this period of time just seem to not exist?
Sort of looking back over historically.
Like carbon dating, yeah. So you're actually mapping the genomes backwards to a theoretical point where
it didn't.
might go back as far as when people were a lot smaller, or when people didn't have X, Y, and Z in our lives.
I think that's a very interesting question, and one we probably need to leave hanging, because I think the session is over. But I think the take home from that is that we are increasingly using sequencing techniques to map the genome. And we are getting to the point where for £1,000 we can map an entire genome.
Yeah.
10 years ago, that took an international consortium 10 years. So it's really improving.